Now, my major professor used to tell me that he'd be happy if I could write a PhD thesis that was as good as Joshua Lederberg's. And that's going to provoke a question, and I won't answer it. But you know the answer. Joshua Lederberg's PhD thesis, which earned him a PhD degree from Yale in 1947, described the discovery of sexuality in bacteria. Immediately after earning that degree, Lederberg came here as an assistant professor of genetics, and here on this campus continued that brilliant work on bacterial conjugation and mutation and transduction that we all know him for, and for which he was awarded the Nobel Prize in 1958, along with Beadle and Tatum. Not long after he discovered bacterial conjugation, Lederberg discovered Jim Crow, and Jim was at the time a young faculty member at Dartmouth. And he was instrumental in bringing Jim here to Madison, for which we are eternally grateful. Lederberg was the founder and first chairman of the Department of Medical Genetics. He launched the new department and then soon after left for Stanford University, where he stayed on as chairman of their genetics department for a number of years until 1978, when he moved to New York as president of Rockefeller University. Dr. Lederberg will speak to us today on 40 years of crossing bacteria.
It does feel very much like coming home again and being old friends or people that I think of more as part of my own family. And what a wonderful pleasure it is to celebrate Jim Crow. As Millard told you, I met Jim 39 years ago. It was the second Cold Spring Harbor Symposium, the one in 1947, at which work on bacterial genetics was discussed. And I found in Dr. Crow someone of unusual perception as to what the fundamental issues were, the experimental needs as well as the theoretical implications of work in a field that he actually knew nothing about, namely microbiology and the genetics thereof. But he had such a breadth of outlook on these and many other questions that indeed was with very great pleasure that one of the most important contributions that I made to the department was to suggest his name in candidacy for the next opening that was going to come up, and he did come in the fall of 1948 to your and my marvelous edification. I'm going to take you back in a historical reminiscence. It has a number of threads to it, and I don't know how successfully I can weave them together. One of them is the Wisconsin experience, which is very intimately connected with Jim's intellectual contributions. He was my closest confidant, my teacher in many areas of genetics that I knew nothing about, and someone with whom I would discuss every experiment, sometimes even before it was done. I've never had a comparable experience among my colleagues since that time. Besides the Wisconsin experience, I'm going to say something about my own personal experience, about how the discovery of bacterial crossing came about, but I would like to put my main stress on the prehistory or the prior history of that subject, of what the actual threads of investigative anticipation there were that led to the discovery of this phenomenon. I'm sure it was in the course of many conversations that I had with Jim in the late 40s and early 50s that I began to concern myself with the historical as well as the scientific dimensions of that discovery, and to ask questions like, why had it not happened before? And in fact, one may marvel that the observation of a phenomenon as elementary as the question of sexual reproduction in a broad kingdom of organisms on Earth took until 1946 to be elaborated. I've become quite discouraged about the development of broad theories of history, even of the history of science, and so I will not really enter into a debate as to whether it's possible to describe a discovery as premature, mature, or post-mature, because that would imply that there is some metric of expectation as to the time at which discoveries ought to be made. But I thought just going through what we do know of the historical background in this area would be of some help in developing a perspective. I'm going to use a number of transparencies, so I think I'll go over to the projector. So I won't need these.
Brought a handmade.
That was to go through the application of the utility of genetic experiments in bacteria, particularly E. coli, that is generically related to species like Salmonella, and to keep bumping into new phenomena that we could not possibly have anticipated. Lysogenicity and lambda, the F plasmid, generalized transduction in Salmonella, specialized transduction in lambda, UG. And then there's a few incidentals, and here I remark on a couple of things that were of particular interest to Jim and myself, demonstrations of the pre-adaptive recurrence of humans that were involved in drug resistance. This is the replicating method, and here is a way of demonstrating pre-adaptive recurrence of such mutations with no technology, no test tubes and pipettes for the elution. Cultures of the retrospective selection of those subpopulations of mutations would be identified to have occurred by virtue of the test sample, that's the SIP selection. And so methodology, the application of penicillin for the concentration of auxotrophic mutants, and that one of the threads of evidence is the mechanism of action of penicillin. The L-form systems of growth of bacteria that have been deprived of their walls. Some theoretical considerations about genetic particles, mostly from microorganisms, and the plasmid concept. Extrapolation of these methodological approaches from bacteria to somatic cells come out very beautifully, and miscellaneous with some other organisms looking at similar phenomena in streptomycetes. And then the last of this litany, and I apologize for those who will be quite meaningless, but I know it's meaningful to Jim myself, that I want to make a remark about the paper that I gave at in connection with the Nobel Prize. The title of it is A View of Genetics. It says almost nothing of the particular experiments that I had done. They're presented as footnotes to the paper, and that's one of the reasons that I'm talking today about the history of those experiments. For some purposes, it might have been better if, as early as 1958, I had collected my thoughts on the historical dimensions of that work, but I was too busy doing it and looking at its implications in other ways. Instead, I used the occasion of the Nobel lecture to address the revolution that in fact had taken place during that year, that it was impossible any longer to think of fundamental issues of genetics without reference to the chemical constitution of the gene, namely DNA, the information described in it, its linear sequence. Today, you will wonder why anybody bothered to make a statement at the time, but believe me, there were still pockets of resistance to this oversimplification of the nature of life, and I thought that was a more important issue to present than the details of my work. Well, that's the Wisconsin experience. But let me come back now then to a little bit of personal history, and then move on to the general historical background. I I mentioned that the experiments that I'm referring to had been conducted, the first successful ones were conducted in June 1946. That makes this very neatly the 40th anniversary. At that time, I was a medical student in the College of Physicians and Surgeons at Columbia. I was not a regular graduate student at Yale. The PhD that Laura talked about was an afterthought, that after the work was all done and tried to find some base for my further career, there was a retrospective adjustment of my registration in the graduate school. What the deep question its legitimacy. I had come to be at Yale at the recommendation of a person whom I owe a great deal, the late Francis J. Ryan, who was the one person I looked up to as my primary teacher in science. He was an assistant professor in the Department of Zoology at Columbia. I had entered Columbia College in 1941, already very deeply interested in biological science. I met Francis in 1942 when he returned from a year's postdoc experience at Stanford, which turned out to be with Beadle and Tatum on the heels of their introduction of mutational analysis in neurospora as a method of studying the physiology of genes. He returned to Columbia to begin his teaching career there in the fall of 1942. I knocked on his door and wouldn't let go until he agreed to allow me to work in his laboratory. And so I was the one who would wash the auger and clean the dishes and do all the other things that were necessary for the work to go on. He taught me neurospora. He taught me a certain amount of discipline. Introduced me to the new wave of thinking about how to analyze gene function through mutational analysis. It's an experience from which I've never recovered. During I went on through Columbia, the undergraduate program, Navy D-12 training, with Saigon, and I worked in the Naval Hospital, which turned out to be a very positive scientific experience, and so I paid my debts in other ways, because there I had the responsibility of looking at blood smears of the 3rd Marine Division that returned from Guadalcanal, and all of them were suffering from malaria. It was my job to do the differential diagnosis between Plasmodium vivax and Plasmodium falciparum. This meant a rather close microscopic examination of what amounts to the life cycle of a unicellular parasite when it does have a sexual phase. It's certainly one of the elements that explains that led me to the generality of sexual phenomena, even among microbes. The important incident that shaped further research was, however, the publication in February 1944, a paper I hope you have already read, so you don't need to look too closely at the title, Avery, Cloud, and McCarty, on the identification of the substance responsible for the phenomenon of serological type transformation in the pneumococcus. They chased us down and identified it as deoxynucleic acid. One of the reasons that I'm speaking a little bit of history today is I've been thinking about it for some few years of the many what I would do misinterpretations of the way in which these publications were received by the scientific community at the time. It is true that there was not an instant conversion by the crucial data that proved to be the ultimate reality. That's a criterion that was not demand or hoped for in the presentation of new and important scientific material, which is not the same as to say that this work was ignored or fundamentally misunderstood or in other ways received inappropriate resistance. I think the reaction of the scientific community was entirely appropriate. There was a widespread recognition that this was a phenomenon that was quite unsettling, its ultimate implications, that the chemical identification of the material responsible for a phenomenon like transformation was one that was deserving of the utmost rigor of demonstration before it was finally and conclusively accepted, but it was given as one of the most important leads that one could hope to find for opening the world of analytical molecular biology. There was another problem about that particular system, namely it was a pure speculation that the pneumococcus dye transformation could be or should be thought about as an example of the transfer of genes. The problem with thinking about transformation as genetic transfer, or one, that experimentally only a single marker had been examined that had to do with the production of the polysaccharide coat around these bacteria. And one could, and many people did, offer a variety of completely legitimate alternative hypotheses as to the biological mechanism that was responsible for that. But more importantly, there was no context in which to think about genes in bacteria. Bacteria hadn't been crossed, there was nothing mentalized, there was nothing about the existence of chromosomes. There were no other data about genes in bacteria that could be connected with these observations on the pneumococcus to make any kind of intellectual system. Well, it was an extremely provocative finding. How provocative it was, I can document my own reading of the article. I had heard about it around Columbia throughout the year, but I'd been off in the hospital on January 20th, 1945. Notes from a kind of commonplace book I had. You certainly can't read the handwriting, I'll read the small text. I had the evening all to myself, and particularly the excruciating pleasure of reading Avery. I call it 43 because that's when I heard it work on the deoxyribonucleic acid responsible for type transformation of the coccus. Terrific, unlimited in its implications. Direct demonstration and multiplication of the transforming factor. I only put this down as counter to the myth that's been propagated that nobody paid any attention to the Avery work and it had inappropriately small influence on further research, nothing further from the fact. I suppose the most important demonstration of that reality is that from 1944 with Avery's paper, which is the first important mention of DNA as having any biological activity of any kind, is only 9 years in that interval between that and the Watson-Crick double helix structure and so forth. And why would there be any fuss about knowing the structure of DNA as a double helix if it were not for the very rapid development of the traditions and importance of DNA? It's a very, very short period of time indeed. Well, my own reaction to that, as you can see from the kind of startle that's reflected in these remarks, was along the lines that I have just mentioned to you, that the work seemed to be the key to the future of molecular understanding of fundamental biological process. But besides the question of the final demonstration of what the chemistry of that material was, and the first reports from the Avery laboratory were very good, but hardly the ultimate final evidence that DNA and DNA alone is responsible for biological specificity. There was also the question of what indeed was the biology of the phenomenon. And I had the sense that if one could only do DNA transformation in a Mendelizing organism, one would have the key to that particular dilemma, and one could bring the tradition of DNA investigation directly in touch with the traditions of Mendelian genetics, and one might wish to have a Drosophila or a maze in which to do that, and we do that these days. But at that time, there were no such systems available at the time. But my thought was that Neurospora might provide the ideal organism for the conduct of such experiments. And so I begged Francis Lyon to let me the first time do some of my own independent experiments in his laboratory, and they were a trial at the transformation of Neurospora auxocopic nutritionally dependent mutants with DNA from the wild type, and using selective conditions of being deprived of the growth factor that the mutant needed efficient way to discover whether there were any transformers. One could get transformation in neurospora, one could study the behavior of the biological trait in what conventionally Mendelian analysis, in fact better than Mendelian, because of the recovery of all the products of meiosis and single acids and so on and so forth. Well, Francis had been working with a particular mutant in neurospora that required leucine, using this to develop a bioassay for leucine in proteins. This was before the days of the amino acid analyzer, biological assay for sensitive terminations. And so that was the culture that I used in those experiments. I did prepare some crude extracts of neurospora. It's quite possible that they had some DNA in them, but that proved not to be the crucial consideration because it turned out that this leucinous culture, when subjected to the severe and stringent selective conditions that were being imposed to discover possible transformants, in fact spontaneously reverted. And so that experiment turned out to be the first demonstration, I'm surprised it was the first, that reverse mutation does indeed occur among these neurospora mutants. Well, that was a little discouraging that we had what looked like a verifiable methodology for the investigation of this result, but there was this artifact that was getting away. It's just as well I didn't try to fix it in neurospora because it took about 30 to 35 years from the date for the development of techniques which we now have that make possible transformation in neurospora. But that discouragement led me to think about another possible tack of responding to the challenge that had been presented by mutants. Or, and that was to re-examine the question of whether one could do crosses in bacteria. One could take the fact that instead of trying to transform an organism whose demendelizing properties were well established, perhaps one could learn more about the genetic system of bacteria and then use that as a framework to understanding of DNA transfer. Here again, well, I'm plotting the record scribbles defining the experiment. In July 45, the thought came that perhaps one could use substantially the same selective methodology that I had mentioned before in the neurospora system, but apply it to auxotrophic mutants in bacteria, mix auxotrophic strains, see if they recombine with one another,